Hello everyone, we are solving question number 7 of GATE 2013. It says endogenous antigens are presented on the cell surface along with MHC class 2, MHC class 1, Fc gamma receptor or complement receptor. So let's see how endogenous antigen are presented on the cell surface. So if you could see here, endogenous antigens are through class 2 MHC pathway. Here you have the antigen that is present inside the cytosol, and if you talk about exogenous pathway, it is class 2 MHC pathway. So for endogenous, it's class 1 MHC pathway. So let's just understand this pathway in details. Endogenous pathway, which is MHC class 1 pathway, is also called cytosolic pathway. It is cytosolic because the endogenous antigen is present inside the cytosol. So there are three steps. First one is processing of antigen into the peptide. Second is assembly of MHC and the peptide loading complex. And the third one is peptide loading and MHC peptide transport. So the first one is basically the processing of antigen into the peptide. So here you have the degradation of the endogenous antigen. So ubiquitin help in degradation. Ubiquitin proteasome pathway for cytosolic protein degradation. So what happened? Ubiquitin have covalent conjugation to the antigen, and this, once there is a covalent conjugation of ubiquitin to the endogenous antigen, it targets the protein to the proteasomal assembly. In the proteasomal assembly, which is a cylindrical shaped catalytic protease complex or 28 subunit for cytosolic protein degradation, it unfolds the protein. First it unfolds the protein and then cleaves the protein into the smaller peptides or amino acid by proteases. So this is step 1 of the endogenous pathway. Then you have step 2, where there is a transfer of peptide by tap proteins. So once the peptides are degraded by the proteasomal assembly, they are transported through tap proteins. So tap proteins are the transporter associated with the antigen processing. They are tap 1 and tap 2. They form heterodimer. If you could see here, they form heterodimer in the membrane of ER to facilitate the selective transport of the peptide from the cytosol into the lumen of the ER. So this is a very selective transport. So once there is a degradation of the peptide, these peptide get inside the ER lumen through the tap protein. So these tap protein transport these peptide inside the ER lumen, where they are presented to the class 1 MHC. So step 2 is peptide being transported by tap protein. So in cytosol, the association of LAMP 2, LAMP 7 and LAMP 10, which is a part of proteasomal assembly, have a cleavage of these peptides in such a manner that these peptides can bind to the class 1 MHC molecule. So within the RER membrane, a newly synthesized class 1 chain, which is associated with the clanexin, until 2 microglobulin bind to the chain. So you have class 1 MHC alpha chain and calnexin remain associated with this. Calnexin is a chaperone protein. It maintains the integrity of the class 1 alpha chain, and once the 2 microglobulin chain bind to the if you could see here the small portion here, if sorry, once this 2 microglobulin bind to the class 1 MHC alpha chain, the calnexin leave the get dissociated from the class 1 alpha chain, and this class 1 MHC molecule Here there is another protein called calrectin that binds to it and maintain the integrity of the entire class 1 MHC complex, where we have alpha chain as well as 2 microglobulin. Okay, so once it is there, then there is a tapsin protein. This tapsin protein actually regulate the binding of incoming peptide to the class 1 MHC. So if you could see, when a peptide delivered by the TAP is bound to class 1 molecule, folding of class 1 MHC is complete and it is now released from the RER and transported to the Golgi to the cell surface. So third step is generation of class 1 MHC peptide. So if you could see here, class 1 alpha MHC and calnexin, they get associated and they maintain the class 1 MHC alpha chain, and once there is a beta 2 microglobulin which binds to the class 1 alpha chain, the moment it bind here, the calnexin get dissociated and calrectin and tepsin come to bind to this complex, and these protein are just maintaining, calrectin is just maintaining the integrity of the whole MHC complex, and tapsin is regulating the incoming peptide which will come directly and bind to the class 1 MHC molecule. So once they are the incoming peptide, which is a part of endogenous antigen, bind to the class MHC molecule, the tapsin and calrectin dissociate from the class 1 MHC molecule, and this class 1 MHC molecule is now ready to exit RER, and through Golgi vesicle, Golgi body, it is ready to present on the surface of the cell. So assembly and destabilization of class 1 MHC, newly formed class 1 chain associate with calnexin, a molecular chaperone in the RER, subsequent binding of 2 microglobulin, release calnexin and allow the binding of chaperone calreticulin, and once the calreticulin maintain the whole integrity of class 1 MHC molecule, tapsin is there so that the specific peptide which is coming through the tap transporter bind to the class 2 MHC, and once the binding is done, they are dissociated from the class 1 molecule, and this class 1 molecule is ready to exit the RER. So once the peptide, we place the tapsin and calrectin bind to the agrotope of the class 1 MHC molecule to form PMHC. That means now it is loaded, the MHC molecule is loaded, and peptide binding provide the stability to the class 1 MHC molecule to allow the transfer to the surface. So from here it is released from the RER, it comes to the Golgi, and Golgi complex, Golgi vesicle take it to the surface, and the class 1 MHC molecule is presented on the surface of the antigen presenting cell. So this is the endogenous pathway, and in endogenous pathway, class 1 MHC molecule is there to present the antigen. So coming to the question, endogenous antigens are presented on the cell surface along with class 1 MHC molecule. So answer here is B.